Hello, I am Dr. Kimmel, and welcome to our secret lab at Cambrian Park. Today, we're going to run a 2-point test cross with one of the organisms that we brought back from the dead. Let me introduce you to the Anomalocaris. My assistants, Sara, Raul, and Shiraz, will help you with getting started on this test. Be very careful though. The species cannot escape, as it will destroy many ecosystems.
Hey, I'm Dr. Kimmel's assistant. We had an issue in our lab this morning when trying to replicate the Anomalocaris. Something went wrong while cloning their genetic information, and we have realized there is a mutation among the specimens that we cloned. Dr. Kimmel suspects that these two mutants traits are linked, so you are here to help us to analyze these two traits using 2-point test crosses. Alright, so first of all, we have here the wild type phenotype from this specimen, which is orange pigment in the cuticles, represented at A+, and the smooth lobes represented at T+. Then we have the mutation, which is no pigment in the cuticles, which is A, and the serrated lobes, which is represented as T. So now we are gonna just check if we have two mated specimens with known phenotype. These are the parental generation, as we can see here. All the F1 progeny from this cross are heterozygous for both traits. So to do the test cross, we will make one individual from F1 progeny with a homozygous recessive. Now, the F2 progeny from this cross will result in 4 different phenotypes, as we can see over here. So now the question is, what are the 2-point test crosses, right? 2-point test crosses are used to estimate the distance between the two or more genes, and the units are normally in centimorgans. The test evaluates two traits that are normally seen to be inherited together. As an example, in humans, the genes for hair and eye color are linked. People with blue eyes often have blonde hair. To determine whether two genes are linked, an analyzing on the progeny should be taken place. To this is done by calculating the recombination frequency.
So we're going to work on some questions, and as new employees, we need your help to get the answers. But don't worry, we will also help you. Our questions are these genes linked? And what is the recombination frequency? What is the distance in Morgans between A and T? Are they coupling or repulsion? And since gene R codes for antenna length, if a second test cross between R and T is done, and we discovered that the recombination frequency is 50%, with 2034 as the total number of progeny, how many of these offspring are recombinants? What are the phenotypes of the recombinant? And looking at the distance between gene R and gene T coding for antenna color, are genes R and gene A usually inherited together? So for the first question, knowing if the genes are linked will help in our understanding on how often these mutations will occur in future generations, and will help determine the location of the genes. So the question being, are these genes linked? I have come to the conclusion that yes, they are. The ratio from the F2 generation shows that since the outcomes are not 1 to 1 to 1 to 1, these genes must be linked.
For the next question, recombination frequency measures the percentage of progeny with a genotype different to their parents. It is calculated by dividing the recombinant individual's number over the total amount of progeny. and then multiply this number by 100 to get your percentage. This is how we got 13.01% of recombinants in the Anomalocaris F2 generation. Now, maybe you're confused about why is this question being derived from the previous one. Well, our answer for the question 2 is the base to solve what is the distance between genes A and T. So, distances in gene maps are measured in either map units or Morgans. 1% recombination frequency is equal to 1 map unit, and also to a 1 centimorgan. Therefore, based on that, we have 13.01 centimorgans. Now, to find the morgans is simple. 1 morgan is equal to 100 centimorgans. Thus, after a simple conversion, we have that the distance between genes A and T to be 0.1301 morgans. Let's dive into the next question. Are these alleles in coupling or repulsion? A good trick to find out is comparing the different genotypes of the progeny. As we know, since crossing over doesn't happen as often between linked genes, most of the progeny will be non-recombinants, which means they'll have the same genotype as the parents. So now, with this information, we can conclude that the non-recombinant progeny are these ones here with the major numbers. This allows us to ignore the mutant alleles from the homozygous mutant parent, because it can only inherit these 2 alleles here. So, if we only look at both the first and third alleles, we'll see how the genes from the other parent are arranged. The first allele in this case is A+, and then we have T. Then we see that 434 individuals had an A mutant allele and T+ on the first and third position of their genotype. We see that these genes are in repulsion when we arrange them one under the other, meaning each gene has 1 mutant and 1 wild-type allele.
We're interested in seeing where these genes are located in relation to other genes, so last quarter we did a test cross looking at the genes for lobe texture and antenna length together, the same way as the test cross done previously. Unfortunately, due to a accident in the lab, we lost most of our data. We remember that there were 2,034 offspring of the cross and that the recombination frequency was 23.5%. From this information, can you work backwards and figure out which phenotypes were recombinant and how many of each phenotype there were? We can figure out the number of recombinant offspring like this, and we can figure out the phenotypes that were recombinant and non-recombinant this way. Looking at the distance between genes R and T for antenna length and lobe texture, and the results of the previous cross, the distance between color and lobe texture genes, would genes A and R usually be inherited together? We can determine this by making a gene map.
Hey, nice to see you. thanks for helping us with this dilemma this morning. Now you have learned about 2-point test crosses. So, welcome to the company. Oh,